This video covers Chapter 15 Notes, which is the last little bit of Unit 5. We will once again look some more about non-Mendelian genetics, environmental effects on phenotype, and chromosomal inheritance. First, we're going to start off by talking about a scientist named Thomas Hunt Morgan. I guess fun aside is that my grandfather was a Thomas Morgan, but not this Thomas Morgan. And the first evidence associating a gene with a specific chromosome came from him while he was doing his research in the early 1900s. His early experiments provided evidence that chromosomes are the locations of heritable factors that Mendel had described years earlier. And he did his research on fruit flies, specifically one called Drosophila, and he used that as his model organism. Fruit flies are great model organisms because they breed quickly and produce a lot of offspring. They only have 4 pairs of chromosomes. When we're talking about them, the phenotype for most common character is what we call the wild type, and any alternative is called the mutant. And when you go to college, if you decide to major in bio and you end up taking genetics, odds are you might end up doing some experiments on fruit flies. They are still widely used in the world as model organisms, as are nematodes and zebrafish. Anyway, Morgan mated white-eyed males with red-eyed females. And when he did that cross as his P generation, all of the F1 offspring had red eyes. And then when he mated, with the F2 generation, he came up with that classic 3 red eyes to 1 white eyes ratio that we would expect to see based on what we know from Mendel. But when he looks more specifically at the sex of those flies, all of the females had red eyes, half of the males had red eyes, and the other half of the males had white eyes. And so his conclusion from seeing that is that eye color must be on the X chromosome. Remember that females are XX and males are XY. And his findings that once again provided support for the chromosome theory of inheritance that says a gene is on a chromosome, and he did find some traits that are on a sex chromosome, meaning the X or the Y. Usually it's going to be the X, though, because Y chromosomes are much smaller and do not have as many genes on them. When we use the term sex in biology, we mean the classification into a group based on a shared set of anatomical and physiological traits, which is largely determined by chromosome. We know that it is more complex than being XX or XY. It does depend on genes on other chromosomes as well, which is relatively new to us. Humans and other mammals have 2 types of sex chromosomes, X and Y. And there are a couple of homologous regions on the X and Y chromosomes so that during meiosis, they can behave like homologs. There are other sex-determining systems. Some organisms use the X0, ZW, or haplodiploid. And here are some examples of those 4. Of course, we're all familiar with the XY system, but you can read more about the other 3 if you are interested. In humans, anatomical signs of sex begin to emerge at about 2 months, and that's going to depend on whether or not a Y chromosome is present. There is a gene on the Y chromosome that is called the SRY gene that is going to be required for the development of testes. If it is not there, or if something is keeping that gene from being expressed, then there will be no development of the testes. Going forward, we will talk about sex-linked genes, which means that gene is on the X or Y chromosome, but you sometimes need to be more specific on designating whether that is X-linked or Y-linked. Y-linked genes, disorders involving the Y chromosome, which would be Y-linked, are only going to affect males. We do know that disorders in humans that are found on the X chromosome are more likely to be found in males than females, and that's simply just because of the number of X chromosomes that males and females have. So here is an example of an X-linked recessive trait, which would be red-green color blindness, which we have already done some practice with. There is also X inactivation in female mammals. Almost all of one X chromosome in each cell in female mammals becomes inactivated during early embryonic development. When that happens, the inactive X chromosome condenses into a compact object that we call a bar body. Most genes on the inactive X chromosome are not expressed, but it is going to be reactivated in the cells that will give rise to eggs, because we need to have both of those to ensure that all of the gametes have a functioning X chromosome. This inactivation is going to occur randomly and independently in each cell during embryonic development. So that means that just because one gets activated in one cell, that doesn't mean anything about any of the cells surrounding it. And so we have an example here, figure 15.8, where we can actually see that it is random. So let's say this early embryo for this cat gets an allele for orange fur and an allele for black fur, and one of those is going to be inactive. And you can see based on the picture of that cat that you can visibly see which one was inactivated, based on the fur color, because there are patches of orange fur and patches of black fur, and we call that mosaic females. It's mosaic because there's more than one thing. Next, we're going to take a look at linked genes, which tend to be inherited together because they are close to each other on the same chromosome. When we are following linked genes in breeding experiments, the results are going to deviate from our expected results because of the law of independent assortment. So instead of seeing those classic Mendelian ratios, we're actually going to see something else. And so once again, we are back at Morgan's fruit fly experiments, and we're specifically here looking at body color and wing size. So the mutant alleles are recessive. Neither of those traits are on the X chromosome, so we're looking at an autosome. The wild type flies have gray bodies and normal sized wings. And the mutant flies have black bodies and vestigial wings, which means they're super small. And the results are summed up next in figure 15.9. So he bred a wild type fly with a double mutant fly. And then he did his F1 dihybrid cross and test cross. And in those offspring, the test cross offspring, he noticed that there's wild type, black bodies with vestigial wing, which is the double mutant. But then he also saw that there were gray bodies with vestigial wings and black bodies with normal wings. And so if those genes were located on different chromosomes, we would expect to have seen a 1:1:1:1 ratio. If the genes are located on the same chromosome and the parental alleles are always inherited together, we would have expected to see a 1:1:0:0 ratio. And you can see the data from his experiment below. He noticed that the flies had a much higher proportion of the combinations in the P generation, which is not what you would expect if the genes assorted independently. So his conclusion was that body color and wing size are usually inherited together in specific combinations because the genes are linked, which again means they are near each other on the same chromosome. Which leads us into genetic recombination. Genetic recombination is the production of offspring with combinations of traits that differ from those found in either parent. And we're going to look at recombination of alleles in relation to the findings of Mendel and Morgan. First, we're going to look at recombination of unlinked genes. Mendel saw that offspring with traits that differ from the parent when he was doing his experiments. So let's think back to our heterozygous dihybrid cross, and we get that 9:3:1 ratio. The offspring that match the parent are called parental types. Offspring that do not match the parent are called recombinant types or recombinants. So let's suppose there are 50% of offspring that are recombinants. We can also say that means there's a 50% frequency of recombination, and that is observed for any two genes that are located on different chromosomes and are not linked. Next, we'll look at the recombination of linked genes. So you may want to go back and review figure 15.9 with that. And this suggested that the two genes were on the same chromosomes because the occurrence of parental types was over 50%, and about 17% of the offspring were recombinants. Because of these results, Morgan proposed some process must occasionally break the physical connection between the alleles of genes on the same chromosome. And guess what? There is. He just didn't know it yet, but that's what crossing over is. So that's why he didn't see that expected, 1:1 to 0:0 ratio. And so here you can see the linkage between those two genes and the example here. And maybe this is more clear to you than figure 15.9. The discovery of linked genes motivated one of Morgan's students to work out a method for making a genetic map, which we also call a linkage map, so we can figure out the order and the spacing of all of these genes. He hypothesized that the percentage of recombinants depends on the distance between the genes on the chromosome. So the closer they are together, the more likely they are to be inherited together. In other words, if there is a higher recombination frequency, the genes are farther apart. The distance between genes we say is in map units, and one map unit equals 1%. This is an example of Morgan and his people's lab. In this example, the observed recombination frequencies between three Drosophila gene pairs, B and CN, was 9%. CN and VG was 9.5, and B and VG was 17%. And so using that information, you can order those and figure out that that is the order of the genes on that chromosome, which you will get more practice with. Alterations of chromosome number or structure causes some genetic disorders. Large-scale chromosomal changes can affect an organism's phenotype and often do. They can be caused by physical and chemical disturbances or errors in meiosis. In humans and other mammals, most of the time, they can lead to spontaneous abortion or miscarriage because that zygote or early embryo is simply not viable because of the alteration of chromosome number or structure. First, we're going to look at abnormal chromosome number, which is going to be caused by non-disjunction. Non-disjunction is an error in meiosis. Either the homologous chromosomes do not separate during meiosis 1, or sister chromatids do not separate correctly during meiosis 2, which you can see here in figure 15.3. And the result of that is gametes with either one extra chromosome or one less chromosome. In the case 2, where the sister chromatids don't separate correctly, some of those gametes will have the correct diploid number. And when we have gametes with an extra or one missing chromosome, we call that an aneuploidy. A monosomy is an aneuploidy where an individual is missing a chromosome. Trisomy means there's an extra chromosome. And one example of a human disorder that's caused by non-disjunction is Down syndrome. Individuals with Down syndrome have an extra copy of chromosome number 21, so it's also called trisomy 21. Some organisms, particularly plants, will have more than 2 complete sets of chromosomes in all somatic cells. So they generally are not going to be as affected as much by these changes in chromosome number. The general term for that is polyploidy, which again is going to be common in the plant kingdom and is important for plant evolution. Sometimes there will be alterations of chromosome structure, and here errors in meiosis or damaging agents can cause chromosomal mutations or breakages. And there are 4 types of those. With the deletion, part of the chromosome is lost. The duplication means that the chromosome has extra copies of a section. An inversion is where part of the chromosome is in reverse order. And in a translocation, a fragment joins a non-homologous chromosome. Deletions and duplications are more likely to occur during meiosis than the other 2. And here in figure 15.14, you can see some picture examples of that. So in part A, which is the deletion, the whole section of D is removed from that chromosome. Duplication, we have a doubling up of the section B and C. With an inversion, BCD gets flipped around so that it's BCD gets flipped. And then with a translocation, part of one chromosome is going to be broken off and attached to another one, which you can see there. Human disorders that are due to chromosomal alterations, or a few of them are listed below. Probably what you are most familiar with is Down syndrome or trisomy 21, which is caused by non-disjunction. Klinefelter syndrome is also caused by non-disjunction, and that is when a male has 3 sex chromosomes, XX and Y. Trisomy X is also caused by non-disjunction, and females, well, obviously they're females, but those individuals have 3 X chromosomes. Monosomy X, which is also called Turner syndrome, is caused by non-disjunction, and those females only have 1 X chromosome. Prudichat is a deletion on chromosome 5, and there is some evidence that some cancers might be caused by a translocation, and one example of that is CML, which is a type of leukemia. Some inheritance patterns are exceptions to standard Mendelian inheritance, and 2 of them are listed there: genomic imprinting and inheritance of organelle genes. Genomic imprinting is the expression of an allele that depends on whether the allele was inherited from the male or female parent. And this is going to occur during gamete formation, where an allele or certain genes is going to be silenced. One example here is a mouse gene that's called insulin-like growth factor 2. Inheritance of organelle genes happens because there are genes that are located outside of the nucleus. For example, mitochondria, chloroplasts, and other plastids in plants have small circular DNA molecules, which we know tells us a lot about how those organelles got inside eukaryotic cells. And so in humans, I guess in most mammals and also in plants, it's going to be the maternal mitochondria DNA or the maternal chloroplast DNA that gets passed on. And that's simply because egg is bigger, has all the organelles. The sperm basically just has DNA. And so those are exceptions to Mendelian inheritance that we also have to be mindful of.